Hello everyone, my name is Ashley Goodnight and this is my Public Review Lecture 1. The lecture I watched was The Physics of Genes and the Promise of Personalized Medicine, given on November 17, 2016 by Dr. Alec Ashkementiv, a professor at the University of Illinois Urbana-Champaign. His work centered around measuring and calculating the force between DNA strands. His team of researchers discovered that the strands made exclusively of guanine and cytosine were less attractive than adenine and thymine strands. From this, they postulated that because heterochromatin, or inactive DNA, is AT-rich, therefore its attraction and compaction form structure to allow the euchromatin, or active DNA, to be read. This project has larger implications for medicine, such as the ability to target only diseased cells, preventing degradation before reaching the target, and ensuring the drug enters the cell. The reason I selected this specific lecture has to do with my interest in bioengineering. Bioengineering at Georgia Tech is offered as one of the electrical and computer engineering threads, or pathways of study. When I saw the title of this lecture and its relation to biophysics, I was immediately drawn in. The field of bioengineering is intertwined with this idea of personalized medicine, with creations like diagnostic devices, monitoring tools, disease biomarkers, machine learning algorithms, and so much more. To begin his lecture, Dr. Ashkementiv gave background information on the structure of DNA. As a biology enthusiast in high school, I both tutored the subject and was eventually elected the student education officer of my high school's bio club. This means that I was familiar with much of the information provided, ranging from the monomer of nucleotides to the karyotypes of chromosomes and the cellular differentiation with epigenetics. What I learned during the lecture was the specifics about methods of sequencing DNA. One method he covered in depth was nanopore sequencing. This works when an ionic current blockable reveals the sequence of the confined nucleotides as the DNA passes through a biological or graphene nanopore. In addition to learning more about the specific methods of sequencing DNA, I didn't know the reason there were so many. He discussed the effort led by the National Institute of Health, which worked toward significantly lowering the cost of the sequencing procedure. In 2014, everyone's collective research efforts led to the test only costing $1,000. This was a significant decrease from the $100 million the first sequencing test cost. Near the end of his lecture, he discussed the developing research into quantum sequencing of DNA. In 2018, researchers coined the term tunneling to describe the phenomenon where DNA uses quantum effects to pass through a potential barrier. This allows the differentiation of the nucleotide bases in sequencing. I found more information on the topic from Quantum Biosystems Incorporated, a leading company in the field. With that, I am concluding my presentation. Thank you everyone for watching.